30, so we should start. Okay, hi everybody, welcome. It's my pleasure today to introduce Kevin Lin, who is an assistant professor at the University of Washington's Biostatistics Department. Prior to joining UW, he was a postdoctoral researcher at the University of Pennsylvania in the Department of Statistics and Data Science with Dr. Nancy Zahn. He completed his PhD at Carnegie Mellon in the Department of Statistics and Data Science under Dr. Catherine Roder and Dr. Jigley. His research focuses on studying cellular mechanisms from single-cell data using ideas from matrix factorization and network modeling. Please.
Thank you. Yeah, so I just realized that date is very wrong, and you can kind of guess where this date came from, but you don't need to get detective work. Yeah, so I'm very happy to talk today about one of our ongoing works. It will slightly be more about the published work, but at the very end, I'll tell you some exciting directions that we haven't yet quite figured out, or we're making progress figuring out. So as the title mentions, this is about multimodal data. I'll tell you a little bit about what single cell data in a multimodal sense looks like. But generally, this project won't really be specific to single cell, even though that's going to be our use case for it. So moving right along. So most of my brain waves and energy nowadays has been devoted to cell biology, where we're trying to understand different parts of the human body in their different systems. So for example, if we were studying the skin system, we can then have this miracle of science fiction-like technology where we can now measure the gene expression of every single cell in a particular tissue slice. So you can imagine a tissue slice gives you rise of tens of thousands of cells, and for every single cell, we can measure the gene expression across my, maybe 2,000 genes of interest, but more globally, like 50,000 genes all at once. And we get a count matrix that gives us a sense of like which cells are doing what, because they might not be one all homogeneous mix. It might be a bunch of different cell types in the skin system, for example. But this data has been around in 2015. It's really been commercialized since then. So in a biological sense, that means this is close to a dinosaur. So in the newer sense, in the last couple of years, we have well, not me, I don't work on the technology end, but the commercialized wet lab people hard at work have been able to gerrymander this technology to also work to sequence much more than just gene expressions. Our cells have many different parts, and the gene expression is just one small part of this entire machine that works at the cellular level. And this is where this word multimodal comes in. And I'll give you a sense of that right here. So the cell is doing many, many things. There's many, many mechanisms going on inside the cell. And you can see, it's kind of not super clear in this picture because you lose the faint outline of a cell. But if you imagine kind of in your mind this faint outline of a cell, and I'm pointing to just the gene expression, there's many other components of the cell that's going on at once. You have some antibody markers. This is how your immune systems figures out what's going on. You have your DNA that's wrapped in these areas of open chromatin regions. So there's many, many things going on in the cell. And a gene expression just gives you one small piece of that entire puzzle. So that's what single cell RNA gene expression offered you.
I think the Zoom audience cannot share screen.
I did not share screen. You are right. Thank you for letting me know, because I was not clueless. So I think we should be all. Now the question is, how do I guess I just minimize this. I don't know how to get this screen not to be literally what you see right here. Hide floating. And then I turn.
I was trying to join the but
Surely this is good enough.
Yeah.
You guys can let me know if this is not. So thank you for that. That'll be slightly bad. Let me try again. Hide video panel, hide floating. Sounds good. So now we have this incredible feat of technology where we can see not just a gene expression, but also something else. this is, for example, just imagine if this projector had a higher quality, you can see the outline of a cell and you see some surface antibody marker. So we can measure more than just the gene expression is the end takeaway. And so because this is multi-data, that means in the same sense of incredible science fiction-like technology, for every single one of my end cells, I get your P1 gene expressions. Those are your counts from the gene expression side. But we also get to see, for example, a different modality, a completely different aspect of biology, in this case, the surface antibody markers. And so, more information is usually better, but these represent completely different areas of biology. And of course, the whole reason we are interested in this is we might ask very broad questions about like which modalities contain which informations, the cell type information, the developmental information, which modality is more responsive to a treatment. How do we know if a cell is primed for getting it? Is it really stored information in the antibody markers or the RNA? So we might ask questions about which information is unique to a modality, or what are the correlation between these two modalities? These are the broad questions as a biologist that we'll like to answer. But I'm going to tell you this more abstractly because we won't focus too much in this talk about the downstream ramifications, the scientific ones. We'll just focus on the statistical task of trying to model these two modalities jointly in the sense that I'm about to describe. So, the thing popped up again.
I'll just
Sure.
make Alex host, and that way this won't keep popping up for you.
Oh,
I think.
I didn't know that. You can do that.
I think that'll work, but let's see.
Oh,
Make host.
4 years of Zoom and I still don't know how Zoom works.
Okay, so hopefully that will stop notifying you now. Let me hide that. And then, yeah, and then.
Okay, thank you. Yeah, it's kind of surprising. I still don't know how Zoom works. Okay, so this is a sense we're going to think about this problem. What is the information unique to a modality, or what is the coordination between the two modalities? We're going to take a more geometric sense to answer this problem. And as with all great ideas, it starts with just scribbling a bunch of plots on the whiteboard. So if you, for example, visualize the two modalities of a particular biological data set, here we're going to visualize all your 10,000 cells in a bone marrow data set where the original authors have painstakingly labeled your different cell types by your different colors. I'm going to visualize the RNA as one plot, the protein as another plot. For those of us who are familiar with high-dimensional visualizations, this is technically a UMAP, so the coordinates are a little bit wonky, but we're just using it as a bird's eye view to give us a summary of what exactly each modality contains. So the same 10,000-ish, 50,000-ish cells, but of course, different types of matrices, so different types of information content. And this is, let's say, a multimodal data set we might encounter in the wild. We might have conceptually this idea of this Venn diagram. There's some information content in each modality, and there's going to be some overlap of where both modalities seem to agree. Some modalities say one thing, some modalities say another thing. So if we have this cartoon of a Venn diagram, let's just keep it very high level at the moment, and you ask me, what are some cell type separation patterns, using my colors, that are, let's say, unique to the proteins? You might look at this visualization, and off the top of your head, you might be like, these, pink to slightly purple shades of colors are my different T cell subtypes. And that separation, if I trust this visualization, seems to be much more obvious in the proteins, in my 97 proteins, than my 500-ish genes. We're just making some observations. This is not a very, solid statement quite yet, but this is some evidence that there's some information in the proteins, not in the RNA. Now, if you wanted to point to the middle part of this Venn diagram, what are some things that both modalities seem to agree upon? Once again, just making some high-level statements. We might notice that that entire mass of purple-pink cells are very separated from my blue B cells, regardless of which modality we're looking at. So there seems to be some weak evidence that there's some coordination between these two modalities that relate your T cells to the B cells. So, okay, these are all high-level observations. And of course, in reality, we don't have any label data because we're trying to make an unsupervised version. We're not going to make our method live and die whether we have labels for all these cell types. So we're going to make this statement in a more quantifiable way, but certainly our inspiration comes from a bunch of plots that start off with. So That's eventually going to be our goal. We want to quantify what is the unique or distinct sources of information in multimodal data. Why are we interested in doing this? So I'll just give you a sense for those of us who have worked with biology of single cell data before. So one is which pair of modalities are the most informative to give me the best totality view of the system. So I just showed you a plot of RNA and protein, but now the technology has exploded. There's many pairs of modalities you can measure at the same time. Which one gives you the most comprehensive sense, the most distinct information in some sense? Between all the choices you have before you spend your NIH precious money sequencing your data. The other one is, now for protein, you have to pick which proteins to sequence. So I might want to be conservative and only sequence the proteins that give me, in some sense, the best bang for my buck. And if I had this conception of how much distinct information each protein contributes, then maybe in a follow-up experiment, I only need to sequence the proteins that give you complementary information that's not contained in the RNA already. So some variable selection type procedure you can envision in your mind. And the third one is the one we'll talk most about because this is the one closest to my interest right now in developmental biology. You might have some scientific motivation on what exactly this distinct and common embeddings really quantify in terms of a biological hypothesis you're trying to study. I'll tell you more about this at the second end of the talk. So these are all the different types of things our collaborators might be very interested in once they have their hands on this incredible data of, once again, the same end cells, but two different modalities. And so we're just going to run with this cartoon right here about I want to figure out an embedding that gives you only the common information between these two modalities, or embedding that tells me these are the axes of variation unique to RNA or unique to protein. Now, I like to say that, yes, my brain is always about cell biology nowadays, but certainly this question can extend well beyond, certainly a biological application. The thing I'm about to tell you is more generally for any multimodal data. So for example, for our friends working on astronomy, you might have images and, radio wave frequencies as, your multimodal data. In neuroscience, you have imaging and EEG images that you might want to figure out what is the thing that these two modalities are, having the same, what are different. You might imagine in social sciences, you survey a bunch of people, but you have a bunch of academic things that you're going to survey and a bunch of financial or demographic information. You want to see how related these two are. So really the thing I'm going to tell you is more broadly for any type of multimodal data, but yes, I'm going to specifically focus on single cell biology. So to get our heads in the right place, let me show you once again, every great idea starts with a bunch of scribbling on a whiteboard. So I'm going to scribble on the screen right now, and I'm going to generate a synthetic data, and just to give us a sense of what exactly are we hoping to happen in the best case scenario. So here I'm going to completely generate synthetically 300 cells. In one modality, I'm going to generate 3 different cell types, synthetic cell types. We're just looking at the first 2 principal components. You can see 3 very obviously distinct clusters. That's one modality. On the other modality, this is simulation, I can do whatever I want. I'm going to make these cells only separable into 2 groups where these light blue and orange cells, they might look slightly brown to you. These light blue and orange cells are aggregated into one big cluster. This modality does not have enough information to separate out these 2 cell types versus this dark purple one. So this is a synthetic data set. I can make however as I please. And now I want to ask the question, let's just pretend I don't actually have these cell type labels, but it's the same 300 cells. They're all perfectly matched between these 2 modalities. Yes.
There is one question in the chat. I think it's a clarifying question, so I'll ask it. Are we generally going to limit ourselves to multimodal data with precisely 2 modalities, or are we going to consider more generally multimodal data with 3 or more modalities?
So this talk will focus on specifically 2. Some of these ideas will naturally extend to multiple modalities, and some of them it'll become quite clear as I talk through that. Yes, some parts I don't actually know how to extend to 3 or more. And while I'm paused, I saw there was another question.
I was just highlighting your question.
Oh, okay. Yeah, so we're going to focus mainly on 2 modalities for now. And by the end of the talk, you'll probably be able to guess which parts will generalize more easily and some parts are not going to generalize so gracefully. And that's, of course, the interesting part of the next evolution of this. But coming back to our two modalities cartoon data set where we don't actually have cell type labels, let's just put the cell type labels back on just so it's an easier plot to digest. I'm going to, guess on what is a good embedding that depicts the common information between these two modalities. And if we stared at this, if this was a classroom setting and we stared at this for like 5 minutes, I would imagine most of us would probably claim if I had to draw a two-dimensional embedding that represents a common information between these two synthetic data, I might draw something like this. Now you'll be like, Kevin, that looks exactly like modality 2. And I would say that's probably the point, because in this particular synthetic data, the fact that I can separate out my light blue from the orange is an ability unique to modality 1. Modality 2 does not have it, so therefore I probably don't want that quality in what I'm calling heuristically as a common embedding. But in contrast, both modalities can separate my purple cells from my light blue orange cells. So I probably, because both modalities have this quality, I probably want that quality in the common embedding. Once again, we're just making some fantasizing statements right now, nothing's too formal. So that's the intuition of what the common embedding might look like if we wanted to draw one in 2 dimensions. And you might be like, Kevin, I probably know how to do this. You concatenate the two modalities together and you just apply PCA. Surely that's going to give me something that looks like this. And that is, of course, one of the first things we tried. And so there's many multimodal dimension reduction methods out there, but they capture what we qualitatively are saying is more like a union of information type idea. So let me show you on our very simple toy example. First of all, let me just review what this idea is. It's as simple as you are imagining it. I just apply PCA. I slap the two modalities together because for the same cells or the same subjects, I see both modalities. So I can just column-wise concat these two matrices together. I rescale them, center them, and then I just run a PCA. And if we remember that the PCA is solved by an SVD, as long as we centered it, then we'll see that it's just a big SVD of my column-wise concatenated matrix. And we'll realize that when we look at this UD matrix, this is my low-dimensional embedding that comes out of PCA. This is really combining all the axes of variation that either came from modality 1 or modality 2. So let's just keep this phrase in the back of our mind, that it's some kind of Frankenstein amalgamation of both modalities together. Because when we apply it to our synthetic data, we'll see that this Frankenstein amalgamation combines all the axes of variation for both modalities. It has this quality that it can still separate my light blue from orange because it took that axis of variation from my first modality. So in fact, most of the dimension reduction methods that make one embedding from multimodal data have this quality where it's not really showing you what I heuristically just showed you as a common embedding. It's really showing you something more like the union of embedding, your union of information. It's trying to summarize all the different axes of variation into one embedding. So all these methods back here. So I put some stars next to some of them because this classification is not as simple as I'm making it seem. But all these methods to do multimodal dimension reduction, you can feel free to ask me about some of them later on, are trying to find in the Venn diagram sense one embedding that summarizes all the axes of variation, which is useful. I'm not saying you should never do it. It's certainly a useful task because it gives you a bird's eye view of both modalities in one single embedding itself. But it's certainly very, very different of a task than being able to separate out my 3 phases of my Venn diagrams into 3 different embeddings, where one of the embeddings tells me what the common information is, what are the cell separation patterns that both modalities agree upon, and what are the cell separation patterns that you can only see in one modality but not the other. So that's to prime ourselves for what's about to happen. Yes.
I've just glanced a bit at Jive. So I thought what they were doing is, you have 2 subspaces, you would kind of find a subspace that's common with them, like that intersection of the subspace. Of course, like they're noisy, so that intersection is usually 0-dimensional, but it is the common subspace.
So
it's not like right?
Yes and no. So at the very, very end, if I get to the end of the question mark ramblings of this project, I'll have some examples of why this field is a little bit funny because no one can really agree upon what common really means. So I'm using common here as a geometric sense. So I want only common in the sense that I only want separation that is persistent in both.
Clustering point of view.
Yes, from a
Looks like a common cluster.
yes.
Okay.
So I will tell you the other ways to think about what a common means when we get to the end of the talk. And this is why, so for those of us who have not seen jive before, it stands for think joint and individual variational embedding or something like that. And you might imagine the word joint sounds like a lot like the word common. And so you might think they're doing the same thing, but they're doing something different. I'll tell you at the end. So, okay, so now onto the statistical method. So I'm going to tell you a little bit about the framework we actually use to solve this problem. So we're going to have multimodal data x1 and x2. This is n by p1 and p2. We're going to assume, because I know someone in this audience is going to ask me, we're going to assume this is already sufficiently low rank so that even though in a literal sense the ambient dimension might be very high, it's already pre-processed to be a low rank matrices because I'm about to run CCA on the high dimensional versions of these low dimensional matrices. So that's why I'm going to get away with this. This projector is not going to do me some favors, but you can see I'm going to decompose x1 and x2 into C, D1, D2 on the cell embedding side and some loading matrices on the RNA side. Those loading matrices, I'm not actually going to talk too much about them because they're just the things I need. They're the transformations of the right singular vectors. It's going to be very obvious what they have to be when I'm done with the talk, so let's not talk about them. We're much more interested in the cell embeddings. Now, one of the elements of my cell embedding, I'm going to call capital C in this like orange shaded color. And I'm hoping to call this the common embedding for two reasons. The first one is that you can clearly see it's being used in both modalities decompositions. But more importantly, we're hoping that the embedding, like in that two-dimensional plot sense, the embedding, the self-separation patterns in this common embedding is specifically the ones that both modalities agree upon. So that's a slightly more abstract notion. I'll formalize this in a little bit. But that's the common embedding. And then we have our two distinct embeddings for either RNA or protein. And here we're going to mathematically enforce that these two subspaces must, in a linear algebra sense, be orthogonal to one another. So this is just to give you a sense of the rules of the game we're going to play. And that orthogonality constraint seems to be a reasonable way to operationalize this word on what do we mean distinct variation in one modality not found in the other.
Like direct sum of two subspaces?
Yes, and this is a literal matrix addition.
Matrix addition.
Yes. So this is literally the sum and product of low rank matrices. The way you read it is the way I'm literally. The only thing that's approximated is this approximation step. And then why are we focusing on a linear decomposition is because some of our downstream applications, I'll tell you a little bit about this near the end of the talk, is that if I just multiply the low rank C matrix with the low rank loading matrices, then I can get the common component of hypothetically every single gene, my P1 genes. So that might be one of the nice qualities of a linear decomposition we might exploit, but that's for downstream analysis. And as I answered for our means questions, we're going to literally add C and D1 and literally add C and D2. And this is where CCA is going to come in. We're going to use CCA for identifiability reasons that I'm going to explain to you in just a little bit. And we're going to enforce that C plus D1 and C plus D2 are exactly the low rank canonical score matrices. I mean, I'm assuming X1 and X2 are already low rank, so I don't need to worry about like, high dimensional spurious correlations. They've already been pre-processed by some other fancy pipeline. And I'm just going to run CCA. I'm going to say that C plus D1 and C plus D2 must be my low rank matrices that I get from canonical correlation. So because we're about to move into canonical correlation, let me just throw on this slide that, as we probably all know, CCA in the first latent dimension is trying to find the axis of variation that yields two very correlated inner products. So we would call this the leading pair of canonical score vectors. And of course, if you wanted more than just a leading pair, you would just do this thing on the bottom, where you can do all the first R CCA components simultaneously. We need to remember that they need to be orthogonal scores within the same modality. That's a constraint down here. But if you just, in a homework sense, derive the Lagrangian of this objective function, you'll also see that it's orthogonal between the two modalities. So the leading canonical score vector of the first modality is not only orthogonal to the other canonical score vectors in that modality, it's also orthogonal to the second, to the R canonical score vectors in the second modality. So you have this, like in some sense, cross-modality orthogonality also that we're going to use to make our method more sensible. So throw this up on the screen again, because we're going to be using it in what's to come. So Z1 and Z2 are my two canonical score matrices. I just ran CCA, nothing super special. And then I'm going to look at one pair of canonical score vectors. So here I'm going to draw the leading canonical score vectors of my two modalities. Each vector is n-dimensional, but they live in an ambient n-dimensional space. And on this n-dimensional space, I'm going to do a decomposition. I'm going to say this n-dimensional vector from modality 1 is going to be represented as a common component, an n-dimensional vector, plus D1, n-dimensional vector also. So this yellow or slightly pale looking color, this is my common component. I'm going to add D1 or D2. So here, once again, we're going to call this a common component because you can literally see it's being used in both modalities decomposition, one from the RNA modality and its corresponding CCA vector from the protein modality. And the distinct component is because the way we drew this figure, you can see that there's always a way to perfectly bisect such that the two distinct components are orthogonal also. Seems reasonable. And then what we're going to do is, if you remember these two plots, I'm going to do this type of decomposition, one per latent dimension. So because I know the canonical score vectors are orthogonal within the modality, but also between pairs of modalities, I can do this decomposition ad libitum for one latent dimension at a time, and it won't affect my ability to estimate the decomposition in other latent dimensions because everything is orthogonal when I look at different pairs of latent dimensions. So I can do a decomposition in latent dimension 1. It will not affect my decomposition in latent dimension 2. They're completely different pairs of subspaces. And this is why we're using CCA to help enforce our identifiability. Now, you might notice there's a few things that seem a little mysterious right now, because so far this decomposition it either feels like it's identifiable or feels wildly unidentifiable. I'm going to tell you that in just a little bit. But once again, we're using the CCA properties to help us do one latent dimension at a time. But if I look at my wish list of qualities I want in my decomposition, you'll see that I can decompose each latent dimension individually because they're all independent. The first latent dimension is independent of the second latent dimension in terms of this, orthogonality constraint. I still have orthogonal distinct vectors because I'm going to construct my C vector so that both my D1 and D2 are perfectly orthogonal. And you might wonder for how much fuss I made about how I'm defining the common geometry. That seems to be nowhere written on the decomposition right now. Right now, you might be thinking that all I'm going to do is bisect every pair of canonical vectors, and then that gives me the first two properties, but I've said nothing about the common geometry, and that is because I've been hiding one extra degree of flexibility that I did not reveal yet until right now. So if you wanted to only get orthogonal distinct components, you'll see that because I'm doing this decomposition one pair of latent dimensions at a time, I actually have a semicircle of freedom to pick where this common component is. For example, it's right here. If I put the common component right here and draw my two residuals, it still would give me orthogonal distinct components. So I can swing this common vector anywhere along this semicircle, and I would still have all the qualities I want. And this is the one extra degree of freedom I'm going to optimize over to pick the best tilt of this common component so that I get all the common embedding qualities that I'm looking for. So let's see how this works in practice. We're looking at one latent dimension at a time. And if I swing this yellow component all the way to one canonical score vector, then you can see then the distinct vector for the protein modality is basically parallel with the canonical score vector, and there's a lot of distinct information that is not shown in the common embedding. That's one thing that can happen. The other thing that can happen is, of course, I can dial this knob in the other direction, and now there's a lot of orthogonal protein distinct information not shown in the common space. So I can just jiggle this around, and of course, the goal now is to figure out what is the best jiggle of this tilt, this common component, such that I get this common property about the point clouds, the density clustering that I'm going to tell you about in a second. So this is the key ingredient we need. This one extra degree of freedom, one per latent dimension, and I still need an optimization procedure to formulate it. So let me tell you the optimization procedure, and then we can, I'll take one or two questions before we move on. So the procedure is, first, I have my two modalities, I have n cells in each modality. It's the same n cells. I'm first, step one, after I pre-process the data already, it's already sufficiently low rank. I'm going to construct the nearest neighbor graphs for each of my two modalities. So I get two graphs. They're the same n cells, and in this particular cartoon, I'm going to draw a shaded pixel for there's an edge between those two cells in that modality. So you can see in the RNA modality, if you apply dbScan or your favorite density clustering method that uses graphs, you can probably get 3 clusters in the RNA modality, 2 clusters in the protein modality. This is a cartoon, so I can do whatever I want. Now I have 2 nearest neighbor graphs. I'm going to then have a procedure to construct a third nearest neighbor graph that is not just my RNA, not just my protein. This procedure takes in 2 graphs and outputs 1 other graph. And the goal of this third graph is that it should encapsulate all the common information between the two modalities, at least from the perspective of an n by n adjacency matrix. So I have n cells, so there's the n rows and n columns. And the simplest way I'm going to tell you about today, which is not the only way to do this, but this is just the simplest version to make the talk progress forward, is I'm going to add an edge as long as there's an edge between 2 cells in either of these 2 graphs. So this is an entry-wise operation. If 2 cells are connected, they have either similar RNA profiles or similar protein profiles, just connect the 2 cells in this third graph that I'm going to tell you about. And you might ask me, why on earth is that even in the right intuition direction? And I would tell you that, for example, if you look at this particular cartoon, you can separate the RNA modality into 2 clusters where the protein combines them into 1 big cluster. So if I aggregate the edges, I'm trying to wash away all the information that is unique to a particular modality. If I just aggregate these two sets of edges, I no longer can actually separate these 2 sets of cells. And that's a good thing because that separation is something unique to the RNA modality, and I do not want that phenomenon, that geometric quality in my target graph I'm going to construct. So this is graph operation. We have other versions in the paper that are a little bit more sophisticated. Happy to talk about offline what are the most sophisticated versions of this. But we're going to proceed forward because this is just a graph. I don't have an embedding yet. So that's my G target. And where the decomposition of this tilt is going to come in is I now know roughly what the common information should look like. And now I'm just going to regularize it using a linear decomposition of the CCA. So here are 1 pair of latent dimensions. I'm going to posit a decomposition into the common and distinct parts. The common embedding itself, if I do this for every single one of my latent dimensions, will itself generate a nearest neighbor graph. And you can imagine what I'm going to do. I'm going to have a graph metric. Let's call this G tau J for adjusting in this iteration. Tau J, the tilt in latent dimension J. I'm going to deploy a distance metric between the graph that I think encodes my common information versus a linear regularized, it's just a linear decomposition of my CCA, but itself generates a nearest neighbor graph. And I hopefully am going to find a decomposition of my canonical score vectors that gives me a G that looks like my target graph. So I'm just going to try a bunch of different tilts until I succeed. And of course, we're going to frame this into an optimization procedure. We're not just going to guess and check. And at the very, very end, then we will have a common decomposition, 2 distinct components, and this common decomposition will best reflect the intended geometric qualities that I did in the first step of the procedure. So let me, because we're statisticians, let me say this first, and then I'll answer your question. Let me just write it down in words, what's going to happen. So step one, I already pre-processed my data. I'm not worried about whether it's high rank or not, because everything has low rank, even if it's high rank in the ambient sense. I'm going to construct a common graph using both nearest neighbor graphs from RNA and protein. That's going to give me G-target. Then I'm going to run CCA on X1 and X2, get my canonical score vectors. And then I'm going to decompose Z1 and Z2 in a sense where I'm just trying to tell myself what the tilts are. And once I have the tilts and the common the canonical score vectors, there's a uniquely defined common embedding. And I'm trying to measure the graph of that common embedding with the desired target geometry. And so that's what the optimization procedure looks like if you insist on seeing some kind of equations to make this a little bit more principled. Yes. All right, so that's overall the procedure. Let me just show you a few examples. I might have, yes. Yes, so this is the R-dimensional optimization. So if I have 30 latent dimensions, this is just a 30-dimensional optimization. The tilt here just means that I'm tilting the common vector more towards modality 1 or modality 2. And there's a unique way that if you tell me the tilts, that uniquely specifies what the common embedding should be. So that's why, yeah, yes. So that's a great question. So we do a practical thing of solving it. This is non-convex. And even though it's non-convex, I'm not even sure how well we can actually solve this optimization problem in like a optimal way. But we have just a procedure that seems to do something reasonable. So this is one of the parts where, depending on how we're encoding what we mean by common or not, there might be better ways to do this in the near future. That is not this anymore. And that's one of the things I'll tell you about.
D is an operation on graph.
Yes, this is my graph Laplacian distance.
How is the graph Laplacian?
So, Grassmannian distance between my two sets of Laplacian basis vectors. Two sets of eigenvectors, I just want to use a distance metric that respects the geometry of eigenvectors. So I'll tell you some simulations first. So first of all, if back to my original simulation, I have 3 clusters right here, 2 clusters right here. Once again, we're thinking of common information in this kind of like clustering sense. I should only call something the common information if both modalities agree on these 2 clusters should be separated. Then if you run most multi-modal 2 modalities embeddings, you get one big plot that separates out all these cell types, all the axes of variation. You pull this amalgamation of axes of variation. That's great. Certainly useful, just a different operation from what we're trying to do, which is to find what is the axes of variation that are shared in common versus the ones that are distinct. So you see in this cartoon, distinct one has still the ability to separate out all 3 cell types because that is some information not harbored in modality 2. But after accounting for the common information, there's really nothing left in modality 2. So that's why modalities 2 distinct embedding looks like a wash. You can't really tell much more additional information that is not already in the common embedding. So that's one thing. Now, another simulation, and then I'll just jump into some of the biology, is we call this a crisscross. So then there's 5 cell types in my simulation. And it's crisscross because even though both modalities can separate my green cells from all the other 4, in modality 1, I can separate out my purple and orange cells from my light blue and black cells. And I'm just going to switch the pairs in modality 2. I can separate out my light blue and orange cells from my, well, now it kind of looks all the same, dark purple and black cells. And so you might wonder what happens when I run amalgamation union of information type method on such a data set or our Tilted-CCA. So when you run consensus PCA, of course, you're amalgamating all the axes of variation. You can separate all 5 cell types perfectly. And when you run consensus Tilted-CCA, because the only thing in common between these 2 modalities is that you can only separate the green cells from the rest, that is what our embedding tells you. And then you can still see in modality 1, the leftover information is separating out my 2 pairs of cell types from the different pair of two cell types. So this is all simulation. Obviously, by the time I show you this, everything looks reasonable. And so now I'm going to just give you a little bit of a sense of what we do on the biology side, but I kind of want to get to the parts I think for this audience that would be more interesting to not say too much about the biology. So let me just say, let me blast forward a little bit that we've been using this for multimodal data sets. It's not quite RNA and protein, it's RNA and something else. And this other modality gives us a hint on how development happens. So for example, for those of us who've seen single cell talks, you see a lot of these UMAPs. And so every point here is a particular cell. And in developmental biology, we're trying to learn what is the order of events that cell types happen, the order that they differentiate in. And so unrelated to us, there's tons and tons of methods that what I heuristically call the layout of the highway. So these are existing methods. You could do this downloading very stably developed software. You basically draw these smooth curves that interpolate the high density regions. And that is how you figure out, these orange cells develop into these, kind of muted gray cells, develop into these light blue cells. This is what single cell biologists do all the time. You can think of this as more of an exploratory method. There's obviously nothing causal in this, but this is a good way to get a sense of like, what is your best guess on how cell types are arranged as they develop through time. Now, this is one thing unrelated to Tilted-CCA. You can do this even for just one modality. You didn't need multimodal data to do this. What we're going to ask with multimodal data set is we know some biological relations between these two modalities, the RNA and this other modality I'm not telling you too much about right now. And I want to know, are these two modalities in sync or out of sync when a time a cell makes a decision? So let me just go back because it's a little bit clearer. This is what canonically we might call the branch point in cell biology, where this is the last cell type before, in an associative sense, not a causal sense, it needs to make a choice on whether to become one of these two end states. And we might ask what is going on at that particular junction in time, because if we understood this junction of time, we could probably develop treatment that targeted, hopefully, and you can hopefully engineer things to coerce cells into one state or another. And so to do that, we basically then ran our Tilted-CCA, and we basically have a metric to measure. Once you have the common and distinct information, you can be like, how much unique information is in one modality not reflected in the other? And so we have some metric that can quantify all these things, and we can see that in the terminal states, the two modalities are in sync, that's fine. And when they're in the middle of making a decision, these two modalities are out of sync. And so I'm not telling you too much about the biological ramifications of this, but that's fine. You can ask me after the talk if you're interested in the biological end. But that's to give you just a little bit of a tease of what's we did on the biological end of this thing. And so we can use this type of logic to understand even more complex systems and things of this nature. Now, in the last 10 minutes, I'm going off the rails. I'm going to talk about some work that is unfinished or either unfinished or unstarted, because even though I just gave a talk about Tilted-CCA, of course, as you probably can tell, there's a lot of things that are, not super ideal. So we're thinking about how to extend this method to the next level. And so I'm going to tell you about some of the directions that I personally am interested in, because, I was a developer of this method, so I kind of know both the good parts and the bad parts. I'm going to tell you the bad parts now. There's no secret. It's already published, so I don't need to worry about, ramifications. And so I'm going to tell you a little bit about how I like to think about these things from a more abstract sense, because once again, even though my brain is all about cell biology, obviously this question of common and distinct information probably extends well beyond cell biology. So this is our method. As I mentioned, these 3 steps. Now, you might point to one of these steps and be like, is this particular step statistically justifiable? Like, is there any sense to make sense of this? So for example, the most common thing you might point to is I'm claiming I'm estimating a target n by n matrix. Now, n is my sample size, so certainly this is not the valid population quantity because this changes as the number of cells grows. So you might ask, is there any hope or any sense I'm thinking of about how we prove that we're converging towards whatever we're defining as the true target graph between 2 distributions? I'll cut to the chase. I have no idea how to do this. Now, I'll tell you why I find this a particularly tricky problem of how do you quantify what exactly is the correct common information between two high dimensional well, let's even do low dimensional distributions. Any distribution that has more than one random variable on both x1 and x2. So this is one of the things that we've been thinking about. So it's difficult to pin down what exactly the population level of our common information is, because in some sense, we should have an intuitive desirable quality of the common information. In the talk that I just told you, it is the fact that cells should only be separated if they're separated in both modalities. It's hard to write that in a population level sense. But even if you could, sometimes it does not yield something that is tractable to actually compute. Like forget about Tilted-CCA. If you started from scratch and you wanted to define this thing and estimate this thing, there are some definitions you can write, but you don't know how to estimate. So just to give you a sense of two of the information theoretic ways to think about what do we mean by common information. These are two methods that have been around for quite a while. The first one is, given two random variables, I'm going to define what the common random variable is I need two deterministic functions, f and g, such that in any instance of the paired xy observation, if I apply f on x and g on y, I always get deterministically the same value. So these are two deterministic functions, f and g, and I want the most complicated w such that I can find the f and g. Some intuition on why you want the most complicated, thank you, is I can always just zero out everything, and that's not a very useful thing. So I want the most complicated, the richest w. The other way is I want the simplest w such that condition on w, both my random variables are independent of one another. And both of these definitions are notoriously hard to think about as you go to higher and higher dimensions. You might be able to get away with this in these original papers for univariate binary random variables or discrete random variables, but this definition breaks down. So this is one reason that we're thinking more about how to define the population quantity of this. Let me jump to the last part, because then we'll just close this talk. So as I stated before, part of the nuance and the interesting part of this field is that no one really can agree upon very concretely. If I just told you I want to learn the common information, I could have substituted this for the word joint, mutual, or shared information, and your conception might not change. So this is like a fault of like almost the English language. When I say a bunch of things, I want to learn the common embedding, that doesn't necessarily define anything specific. So there's a figure right here, but the projector is not helping me. So let me just verbally say an example of this. So let's say I surveyed a bunch of people, and on one side, I learned some academic information. I learned what their GPA was, what their SAT scores are, what classes they took, what scores are their final grades for all those classes. That's one modality. For the same people, I then ask their hobby information. So I want to ask, what sports they play, what Netflix shows they watch, how much hours they cook, do they enjoy cooking? So these are my two modalities. One way to sensibly define the common information is the relation between these two modalities. So if I wanted to make a map of all college students and I ran this survey, the way I can map out all the students is saying, oh, there's a correlation between, let's say, how much you enjoy STEM classes and how much you enjoy like shows like Breaking Bad or Star Wars and things of this nature. So this is one way to build a map of all the students where you say, I want to only have the axes of variation that both modalities, my academic information and my social hobby information, seem to align with one another. That's one sensible way. The other way is what I call the house of representative ways. I'm going to construct a low dimensional map of all the students by just taking the largest variance in either of the two modalities and just slapping them together. So on one side, because most of the students here at UW study biology, so one side is all your biology grades, and in the other direction, it's just like, oh, which sports do you like? These have nothing to do with one another, but this is another way where I sensibly map everything into one latent dimension space. And so I can also justifiably call this a common embedding. So you can see there's many different ways you can articulate the word common, and it really depends on how you're going to define what exactly common means. You want a correlation-based way or a prediction-based way, where I'm just saying a prediction-based way is kind of like your house of representatives type thing. You just elect the biggest sources of variation, and then you just slap them together. Now, the other thing that is equally interesting, so this is more about how do you identify the subspace you actually care about. One thing is you can say, I want my distinct subspaces to be orthogonal to the common subspaces. So in a literal sense, Jive is doing this option right here. So all my distinct information is orthogonal to a common information. Another choice is what we're doing. We're saying these two distinct components may or may not be correlated with common information, but I'm going to specifically preserve that the two sources of distinct information is orthogonal with one another. So you can see different choices can yield very different types of embeddings depending on what you want. And of course, the third option, which is notoriously hard, this is why there's very few papers that actually try to do this, is all three embeddings are orthogonal to one another. And so you can see in Tilted-CCA, where even before we talk about the math, we set out to do option 1 over here, this correlation-based embedding strategy, and option 2 down here, where we're saying the two distinct components are orthogonal. And had you chosen any other of the 5 combinations, you will naturally get a completely different embedding that will be useful in its own right, just a very different type of embedding. So, this is part of the things we're commonly thinking about. So just to wrap up in the very last slide, so here is a method that I described that can separate out the shared information from common information. We use it in cell biology for some applications that I've loosely just teased. But we're passing this question of geometric separations into a matrix factorization to help us do the estimation part. And specifically, we're building upon Tilted-CCA to tilt the common embedding, just to make this whole estimation procedure maybe not optimal in any sense because we don't have theory at the moment, but at least tractable. What exactly is estimating, as I mentioned, we don't even have a population definition. And I'm not convinced anyone really has a population definition of what a common random variable is. So, that's not a fault of ours, but, this is, the direction the field is probably going to move in. And of course, the first theoretical question that we're very interested to study is what on earth are we trying to estimate? There's tons of ways currently there's tons of multi-headed VAEs are claiming they're estimating some common embedding. There's tons of ways. What is the actual population? What are you actually converging towards as you get more and more samples? And of course, on the biology end, we're going to apply this method to understand some of the granularity in microglia reprogramming. But that part I'll save for another talk because that will be more biology. And with that, thank you so much. So this is the people I'd like to thank. Nancy Zhang is my partner in crime in many ways because she was my postdoc advisor. Hao-Ye in the audience has been helping on the deep learning side of things to scale this method up to overcome some bizarre artifacts that we'd rather not have the currently linear Tilted-CCA version have. And we have 2 wonderful biological collaborators, the wet lab people at UPenn, who's helping us fuel a lot of the questions we're asking in this particular talk. And with that, thank you so much.
Thank you very much. Any questions from?
Do you located at this head to construct the camera?
I think the question was, you can repeat it if I'm wrong, is if I use Euclidean distance to construct the target graph. Yes, so here we, so the procedure is, once again, let me go to this slide. We get 2 low rank matrices. So just imagine then I just take the leading left singular vectors of x1, form a nearest neighbor graph using just k nearest neighbor distance in a Euclidean space. The leading left singular vectors of x2, same thing, k nearest neighbor graph in a Euclidean space. And then we're going to construct our G target combining these 2 graphs. So yes, spiritually, everything is in a Euclidean sense. Yes, so in this current framework, one of the core weaknesses, which we've been able to get around a little bit well, but I do wonder if we've just been lucky or this really is more complicated than we realize. We're putting a lot of faith into the pre-processing. So the reason we've been able to handle 40,000 genes and 200 antibodies and sample size goes out is not as high as we hope is because we're leveraging the latest and greatest ways to denoise my RNA modality and my protein modality. And I'm hoping this is the leap of faith we're going to take, is that after I denoise it, then Euclidean distance is sensible. Now, obviously, that is usually not the case in the absolute sense, but in practice, we've been able to get away with it. Like, it has not been the, of all the things to improve, this particular part has not been the most damning one. But yes, of course, we would like to avoid it if we could.
I hope that some of your years to generalize very easily to high-dimensional data, some of them wouldn't. I'm not quite sure which one is which. So maybe outline what kind of problems have with high-dimensional.
Yeah. So we completely outsource all the high-dimensional problems because we're just assuming you have a separate procedure, hopefully designed by someone else who are more sophisticated in analyzing bioinformatics in our case, that can just reduce it, that it's still an ambient high-dimensional data set, but it has a low-rank structure, that exactly low-rank structure now. So I mean that's the literal answer. Spiritually, the more useful answer is that we do hope that this method can filter out your noise that might still exist in your data set. And you might wonder, and we have some simulations that kind of show this is sometimes doing the job, sometimes not. If you look at these simulations, if you have a very noisy embedding, this will naturally affect your ability to estimate an appropriate amount of common information. So this is an exact decomposition in the sense that your noise still is preserved somewhere. So you're hoping that your pre-processing can really throw away all the noise as best as you can. So this part is, we're still thinking about can you either denoise at the same time or filter out some of the additional noise even in this step. And then when it comes back to the original question about having more than two modalities, it's really in this tilt part that will be a little bit challenging. So there are ways to do CCA in multiple modalities. That's not the hard part. The hard part is figuring out then how do you use this idea of a distinct component when you have three different canonical vectors. That's really the bottleneck, not the fact can you do CCA with three modalities.
Couple questions.
Mhm.
The next one. So the first one is going back to like the CCA optimization problem. I guess part of me thinks like, okay, if anyways, I'm not going to be able to solve the optimization problem exactly. Why can't I just, when I'm putting CCA formulation, stick this whole thing of like C plus D1, C plus D2, D1 and D2, their inner product is 0. I go after that optimization problem. And why am I doing this like tilt thing?
So the practical answer is we tried that and we couldn't get it to work. Okay. Not a great answer, but that's the practical answer. So this just made the optimization problem a little bit more manageable because now there's literally only R parameters and I know how to exactly adjust this parameter. So that is more of a practical reason. Abstractly, there's no reason I can't do what you suggested. It's more just like currently, the search space is a little bit too wild that I found it more easy to go through CCA because it gives nice identifiable conditions. One latent dimension is orthogonal to another pair of latent dimensions. So this just made for my thinking a little bit more tractable. But there's no reason your method is not good. No. It's a reasonable idea. We just ran into a wall, as most things. Yes.
So if you go back to kind of where you have the two matrices with the green and the just the basic formulation of the model. So I'm a little bit rusty on this, but so that when I see that, it kind of reminds me a little bit of Docker decompositions, maybe with some constraint because essentially you have the C common piece in there, but it really looks like you're trying to do some sort of Docker decomposition. So what is the relationship between Docker or I don't think that Paragraph is
so
right comparison here, but
I don't know specifically for Tucker because this isn't a tensor in the sense that P1 and P2 might be different. Now, so I can't answer your question directly, but I can tell you that initially we did think to pass this whole, like on the surface level, if I started my research question from here, I can pick what data set to work with. So there was a route in the very beginning just to frame it as a tensor decomposition problem. From the very, very start, I just somehow mapped my P1 genes into P2 antibodies. So now my tensor is N by P2 or something. And then just do everything through a Tucker decomposition. And so the reason we stopped that is because just because of the biology. We lost a lot of information translating this into a tensor. That's not to say in other settings this won't be a good idea. It's just in biology for the single cell we were caring about, we didn't know how to do this. But it's even if you could, I have no great answer on what the real difference is because we just haven't been able to coerce ourselves into a setting where we can reasonably compare these things. But that's certainly a very interesting question. Oh, well, I'll still be here, so you can you can. No. Well, thank you.